Alrighty. Oh, I was testing this, and now it's making weird noises. There we go. All right, so we're gonna try this again. I recorded this at school already, and fun fact, it does not record sound, so I just talked to myself for half an hour for no reason. So that was fun. So we're gonna try this again. Our final section of notes is on mutations today, so we're just going to look at what happens when it goes wrong. These notes are in the note package that you guys got yesterday with the biotech stuff. And yeah, we're just going to go. The whiteboard is already on the whiteboard. I don't have a digital whiteboard for you guys, so I'm not going to be drawing anything today. So hopefully that'll save us a little bit of time. And if you hear weird noises, it's probably my dog or cat or a child. So just ignore it. Okay. And as always, I apologize for these videos because I know they're not your favorite things, but it is what it is. So it'll be fine. Hopefully the sound works when you guys listen to it at school. All right. So any change that we have to a base sequence in your DNA, so if we change any of the letters like A's to G's or C's to T's, that is considered a mutation because as we've been talking about, DNA, again, is used to make RNA, and RNA will eventually be used to make proteins. And so the code that we have in our DNA is coding for very specific protein building blocks. So if we change those letters, we could end up changing what the protein is supposed to look like completely. So yeah, anything that we change in the DNA is a mutation. And even though sometimes mutations get or have kind of a bad reputation for being bad and that mutations are bad things, they're not all bad. If we didn't have mutations, we wouldn't have variation in our species and in any species. The fact that we have different eye colors is because of mutations. The fact that we have different hair colors is because of mutations. So mutations help to just make things a little bit more spicy in the different species that we have on our planet. It also helps to create some of the different species on our planet as well. So mutations are not always a bad thing. And as you guys can see on the board, I have mutations written beautifully in like black marker or something. And then coming off of it are 3 little lines. We have beneficial mutations, we have neutral mutations, and we have harmful mutations. These are our general categories. These are the types of mutations you guys have already heard about in Bio 20, I think in Science 10, potentially also in Science 9. So usually we just categorize them into, are they going to help the organism? Specifically, are they going to help its fitness, which has, again, nothing to do with how strong it is and whatever. It has more to do with, does it have the chance to have babies? What is it gaining to increase its likelihood to reproduce? So beneficial mutations are increasing the chances of having reproductive events. Neutral mutations, there's no change. So you're not reproducing more, you're not reproducing less, you're staying the same because it's not actually doing anything. It's neutral. And then harmful mutations are the ones people usually think about. They're the ones that hurt you and are harmful to you and usually decrease your chances of reproduction because in some cases, harmful mutations harm you to the point that you are dying. And if you are dead, you are probably not having babies. And by probably, I mean you are not having babies. And so mutations are more complicated than just the three basic: it is helpful, it is harmful, or it's meh. So we're going to be delving into quite a few of them today. We have two other kind of subcategories of mutations. You have your point mutations, which I will go into 3 different types of those. And we have our gene mutations, and I will go into 5 different types of those. So lots. And again, just brushing the surface of all of the things. So the 3 types of point mutations that we have are silent mutations, missense mutations, and nonsense mutations. Silent mutations are exactly what they sound like. They are silent. They would also technically be considered a neutral mutation because they don't have any impact on the organism. In this case, we're not changing the amino acids that we are eventually planning to make from the DNA. So even though we may have changed a letter, we're still making a histidine amino acid in place of a histidine amino acid. So the overall protein that we're going to get from this is going to stay the same. A missense mutation, I like to think of it as a mistake. So something has happened, and instead of making one type of amino acid, we end up making a different type of amino acid. So in this case, instead of making a histidine, we end up making a threonine. Coming up with amino acids in my brain is kind of hard right now. So because we're changing the amino acid, so the actual little Lego piece in that protein completely, that's going to change the overall structure of that protein. And in some cases, that can be a good change and can lead to a beneficial mutation, but it can also be a bad change and lead to a harmful mutation. One example, I think it's an example of a missense mutation. Now I'm doubting myself. Yes. And I'm having a stroke. Just give me a second. One example of a missense mutation, and I'm sticking to my guns with this one, is sickle cell anemia. Okay, we have sickle cell anemia where you have your red blood cell that is a nice, beautiful little circle. Okay, it has its hemoglobin, little protein in the middle that's capturing all of the oxygen molecules and, oxygenating your body and helping to keep you alive for longer than 7 seconds or whatever it is. And we like hemoglobin. Hemoglobin's great. We like it to be like the quaternary protein that it is and just kind of do its job. But sometimes we have mutations where we have a slight shift, a slight change in that protein structure, which again can be because of just one change in the amino acid. And when that happens, we go from this beautiful circle that we love to a sickle shape. So sickle cell anemia, again, is where you have a mal-shaped red blood cell, and that mal-shaped red blood cell comes from a mutation of the hemoglobin protein where it doesn't form properly, and because the protein is misshaped, that causes the entire cell to be misshaped. And so again, mutations can be multiple things. So silent mutations can also be neutral mutations or are neutral mutations. A missense mutation can become beneficial or harmful. In the case of sickle cell anemia, it can kind of be both. So sickle cell anemia is very common in Africa, in a lot of African countries. There is also a high rate of malaria, which is a parasitic infection you can get from mosquitoes that you kind of get stuck with forever and can eventually, in some cases, lead to death if it's not treated properly. So, with sickle cell anemia, because the hemoglobin is shaped funny and because the red blood cell is shaped funny, people with sickle cell they're immune to malaria. So, they can't get that parasitic infection because their cells are shaped weird. And so, in that sense of it, it would be a beneficial mutation because those individuals are benefiting and potentially increasing their reproductive chances by not getting malaria and dying, which is awesome. But on the other side of the coin, it again can be seen as a harmful mutation because with those sickle-celled red blood cells looking kind of like these weird little C's, these little C's can hook onto each other and can potentially lead to clotting issues. And depending on where clotting issues happen and how bad the clots are, that can also lead to potential death. So this missense mutation situation where you're changing one little Lego block can cause a huge change to the overall protein. So silent mutation, nothing's happening. You're keeping everything the same. Missense mutation, you're making a mistake. One amino acid for a different amino acid. And then a nonsense mutation, think no and stop. Stop means no. If someone's telling you to stop something, they mean that they don't want you to do something, okay? So no, stop. And in that case, our amino acid that we're supposed to make, which I think again is histidine because it's late and I'm getting tired and I can't remember things, the histidine is going to turn into a stop codon. And so during this process of translation where we're making that nice protein chain, we're stopping early, which means that the protein that we're supposed to make isn't being made. And a lot of nonsense mutations can end up becoming lethal to that cell. And hopefully it's just impacting one cell, the cell dies. And if you're made up of more than one cell, that's not usually a big deal. If you're a single-celled organism, then it's a huge deal. But yeah, nonsense mutations aren't usually great. All right. So I have this strand of mRNA, which again, mRNA comes from DNA, and mutations can also happen in your mRNA because of the DNA, sorry. So the top strand is our normal strand. So we are supposed to have an amino acid sequence of methionine, asparagine, I think, proline, and threonine. So a nice 4 amino acid protein chain. Silent mutation, the example here. I'm looking at which screen. We've changed this adenine base to a uracil base, which not great, but if we looked up these codons in our codon chart, we still end up with the same amino acid. So threonine is changing to threonine. So it's not doing anything. We have the same 4 amino acids in a row, which means we're going to end up making the exact same protein. For our missense mutation, which again, we've made a mistake, we have now put a cytosine in place of the adenine. And in this case, we are completely changing that amino acid from a threonine to a proline. And so in this case, again, we're going to still make a protein, but that protein is going to have a slightly different shape. And these slightly different shapes of the proteins are going to impact their function. And then in this case, we have 2 spots that are being impacted. So we have a U and we have an A being changed from an A and a C. And UAA is one of our stop codons. So instead of making a 4 amino acid long protein, we only have a 1 amino acid long protein. And this is probably not a functional protein, or definitely not functional for the purpose that it was supposed to be. So silent, nothing changes. Missense, we just change 1 amino acid or 2. And nonsense, we stop the process early. So those are our 3 point mutations, where we're just focusing on single kind of sections or single points on the DNA. Now moving on to our gene mutations, which again, there's 5 of them, but 3 of them kind of go together. So It's not that scary. Alrighty, our gene mutation examples, the first two, I hope are pretty self-explanatory. You have an deletion mutation, so you are deleting things. You're getting rid of some of the letters. Insertion, you are inserting letters, so you're adding bases to the DNA. And by adding or deleting bases, you are going to cause what is called a frame shift mutation, where you're changing the reading frame and the amino acids that are going to come out of that portion of DNA. If you're inserting things, you're adding to the reading frame, so you're kind of shifting everything over one, which means everything as in you're shifting all the bases over one. So you're going to end up changing all of the amino acids that come after that insertion that has happened. Same with the deletion, you're going to take something out, everything's going to shift backwards, which means that again, the letters are going to be out of order. You're going to end up making different amino acids. Translocation is where you take DNA from here and you move it over here, because why not? Because we can. Typically, this is happening when you have crossing over between non-homologous chromosomes. So at the start of the year, we talked about prophase 1 in meiosis. Your homologous pairs come together. They're homologous because they're the same, as in they have the same genes. They're the same size, like chromosome 1 pairs with chromosome 1. They can hold hands. They can rip each other apart and exchange DNA, and everything's fine. Because again, you're changing hair color for hair color, or hemoglobin for hemoglobin, okay? With translocation, usually it's like chromosome 1 decides to pair up with chromosome 7. And they look at each other across the nucleus and they're like, oh my gosh, let's be friends forever. They come together and they do the same thing. They cross over, they hold hands, they rip each other apart, they exchange their genetic information. But the issue is chromosome 1 and chromosome 7 don't have the same genes. So we might be changing a hemoglobin protein for a keratin protein, so the things that make up your hair and your nails. That's going to make some wonky things happen, okay? And then inversion is where you take a piece of DNA and you just flip it. So instead of it going TTAC, it's now going CATT. So it's the same letters, just backwards. Okay. And then the next couple slides, I have pictures to show you guys this, because I know that it's good to see pictures. And I also can't remember how much of this you guys actually have to write down, so I'm not going to wait very long this time. But we're going back to that original piece of DNA that I had for my point mutations. So methionine, potentially asparagine, proline, and threonine as our amino acids. We have a deletion mutation example and an insertion mutation example. And the other thing that you guys need to look at, and what I've already said, is that mutations can fall under multiple categories. So they can be beneficial, neutral, harmful. You can also have gene mutations that happen that lead to different point mutations. So in this case, our deletion mutation, we're removing something, and as you can see, we have a threonine amino acid up here, and we now have a histidine amino acid down here. So we are changing the amino acid that's supposed to be there. So I want you to take a second and I want you to write down what kind of point mutation that is when we change the amino acids. Okay, now the insertion mutation, you're going to do the same thing. In this case, we've inserted a U. We now have UAA. That is a stop codon. So we are going to stop this process before it gets too far. And methionine, again, is going to be the only amino acid. This is probably not going to be a functional protein. So what is it called when we stop translation too soon? Okay, I counted to like, I don't know, 7 or 8 in my head. So drum roll. The first one is a missense mutation. So you've made a mistake. You have changed and swapped out your amino acids. So might not be a huge problem. Might be a huge problem. We don't know. And then our insertion mutation one, where we have the stop codon, that again is your nonsense mutation. So we're saying no, we are done with this translation process. And again, these are probably not great mutations to happen. So it's important to know, again, that some of these gene mutations, even though they're under that gene mutation category, can also be point mutations. And again, I can't remember if I have you guys labeling these or if you even have these pictures, because I made the notes last week and last week was a long time ago. So this frame shift mutation picture, this is your normal translation. So with this specific sequence of RNA in this case, which again came from DNA, we have these amino acids, so threonine, arginine, threonine, alanine, and tyrosine. When we add a base, so in this case we have an insertion at this point, we are shifting everything 1 over, and that again is going to drastically change the protein that we're going to make. The only amino acid that really stays the kind of close to where it was is arginine. It's now first in line instead of second in line, but we now have a glycine, a leucine, and a histidine that didn't exist before. So very, very, very different protein that we're going to be making. If we look down at this deletion one, we're taking, it looks like an A away. So this A is disappearing down here, I think, or a G. Something's disappeared. We've gotten rid of something. And now none of the amino acids are the same as the original. We have a glutamate, we have an asp that I can't remember what it is. We have a cysteine, we have an isoleucine, we have a proline. None of those amino acids were in the original translation in that original strand, which means that this protein is definitely not doing what it's supposed to be doing. Which, when proteins are doing the wrong thing, that's not, again, usually a great thing. Inversion, this is showing you that we're taking this piece. Sorry, my mouse is not working. This piece, we're just flipping it on its side, completely upside down, and putting it back in the same spot. So the, where is it? The GACTGCCGT that we have on the bottom here is now on the top, and it's backwards. So the GAGTGGGT, that is over here. Sorry. And then translocation, we again are taking DNA from one spot of the genome, moving it to a completely other spot. In this case, you have chromosome 4 and chromosome 20 deciding to get all cozy together and then rip each other apart. And not a great thing, because usually, or at least one example of a translocation mutation where we have this abnormal crossing over can lead to different types of leukemia, which is like bone marrow or blood cancer. So not great, kind of harmful, kind of not awesome. So we want our homologous pairs to match up, not our random pairs to match up. So those are our different types of mutations. I know that was a lot, and I'm sorry, there's a little bit more. We just have to finish with how we can now use mutations. Sorry, I'm going to tell you how this happens first. But then we're going to get onto mutations and how we can use it to kind of map out evolution. So I'll stop talking so you can write this stuff down. Okay, so before we get into the evolution aspect of this, what causes mutations? Where do they even come from? Can we make them happen ourselves? The answer is no, we cannot. Mutations are random. They just happen. We have no control over them. We can't just create a future of X-Men where we're like, hey, I want to have laser vision and I want to control the weather and I want to fly and I want to turn invisible and I want to do all of the different X-Men kinds of things. We can't do that, okay? We can't say I must have this mutation right here. What causes these mutations is random events, specifically spontaneous mutations that occur during DNA replication and induced replications that happen from external sources, like chemicals. So you have exposure to certain chemicals that are bad for you, that can lead to mutations, or radiation, like getting too many X-rays in a certain area can lead to the development of different types of cancer, or exposure to UV radiation without proper, sunscreen type situations, you can end up with skin cancer. So induced mutations where you're looking at stuff like that isn't usually going to create something that's good. It's usually going to create some sort of harmful mutation. Spontaneous mutations can create whatever they want because it's DNA replication, and sometimes DNA polymerase 1 messes up and doesn't spell check properly. Okay, now we are moving on to the evolution part. Okay, so mutations are needed. They're necessary for there to be change happening on our planet, especially when we're looking at asexually reproducing organisms like bacteria, where their whole job in life is to just divide and create identical copies of themselves. The only way we can have changes crop up in bacteria is by mutations happening. And it's important to remember, too, that with mutations, if I have a mutation and I pass it on to my kids, that isn't impacting the entire human population for a long time. If that mutation is a good mutation, and if my kids have a bunch of kids, and if their kids have a bunch of kids, and if their kids have a bunch of kids, and so on and so forth, and that mutation continues to be passed on, then over many generations, we might start to see a change occur in the population, especially if natural selection says, hey, this mutation's pretty cool, we're going to let this build up and maybe lead to the development of a brand new species, if it is a specific structural type of mutation, I guess. But with mutations and the fact that we can now sequence DNA and we can compare DNA between different organisms, we can also look up and compare these mutations between different organisms. So if I have similar mutations to a chimpanzee, or if I have similar mutations to a banana, we can kind of see how closely related we are to each other. And this just helps scientists come up with a better idea of, just how things are working and connected. And up to this point, we've been talking about nuclear DNA, so the giant amount of DNA that we have in our nucleus, the little brain of our cell. And for humans, that is a huge amount of DNA. Again, 2 meters worth of DNA, billions of base pairs worth, and that's a really, really difficult thing to work with. So throughout research, we have now found that our fancy, wonderful, amazing organelles called mitochondria are more than just the powerhouse of the cell. They are more than just making all of the ATP and doing all of that awesome stuff. They do also have their own DNA. And so now we can use mitochondrial DNA, which is a lot shorter and a lot easier to work with, to look at some of these evolutionary relationships. We also found that chloroplasts also have their own DNA. So our little photosynthesis factories that are just pumping out sugar, these two awesome organelles that we spend so much time, not really, but a little bit of time talking about in Bio 20, with all of the important things they do to like run the world, they also have DNA that, again, are smaller, they're nicer, they're more compact and fun, and easier to study. Mitochondrial DNA, you'll most likely get a question on the diploma. I haven't seen chloroplast DNA before, but it's good to know that it's there just in case a question like that comes up. And then usually with mitochondrial DNA, the questions that I've seen about it typically just say, where does it come from? And it's important to know that usually, not 100% of the time, but usually mitochondrial DNA comes from your mom. So it is passed along the maternal line. So when we're looking at inheritance patterns and when we're looking at evolutionary linkages, we're just looking along the mom's line of that family. The reason this happens is because during the fertilization process, where you have an egg cell just bopping around, and then you have a little sperm cell come up and fertilize the egg, the tail of the sperm after fertilization falls off and just kind of floats away. And the mitochondria for a sperm cell is found only in the tail. The head portion of the sperm is just the nucleus, so just the DNA and some enzymes that help it to burrow into the egg to allow fertilization to happen. So sometimes, rarely, the tail will get stuck in when the whole fertilization thing happens. And so some people do have their dad's mitochondrial DNA as well, but that is more of a rare case. So something that most of you, if you wanted to, could give your mom for Mother's Day, because Mother's Day is a great time to say, hey, Mom, you're cool. Give her a card with a mitochondria on it and just say, hey, Mom, thanks for giving me something Dad can't. And then just walk away and just see what happens. And not surprising to any of you, I'm sure, I 100% gave my mother a card that looked exactly like this. I was just like, here's a mitochondria, Mom, thanks. And she just kind of looked at me and was like, cool, that I don't understand, but I had fun. So mitochondrial DNA comes from your mom. Almost done. I have like 2 slides left. So looking at evolution as a whole and trying to figure out how all of the life on our planet is connected is definitely something a lot of scientists are working on. But lots of people are also like, hey, humans, we're pretty cool. Like, how did we do the things? So we have 2 theories on how people on our planet came to be where we are on our planet. So how we ended up with our current distribution patterns that we have. How did we end up in North America, South America, Australia, Europe, Asia, all that fun stuff? And the 2 theories we have are the multi-regional theory and the monogenesis model. The multi-regional theory says that we moved out of Africa because the origin of our species was in Africa, and then we all just said, okay, bye. Like, it was nice knowing you, but I don't want to live here anymore, so we're just going to wander around and move all over the planet. And by chance, we just happen to all evolve and change into Homo sapiens around the same time, which scientists think was about 2 million years ago-ish. So we left Africa as the species Homo erectus, so we were hominids that were able to stand up, and we all turned into Homo sapiens dispersed around the planet, which, if we follow evolutionary theory and if we're following natural selection, the environment has a big, big impact on speciation and how things change. So the fact that everyone was able to leave Africa and live in like the northern hemisphere, the southern hemisphere, over here, and we all ended up turning into the same individuals. Some people have a hard time wrapping their brains around that and accepting this theory. And so scientists said that the groups that left could interbreed because our genes were close enough that we could still have babies together, and those babies could have their own babies. And then eventually over time, our genetics just kind of got close enough to each other and we were able to all change into the same species. So that's the first one. More scientists follow and believe in the monogenesis model, which says we moved out of Africa 2 times. The first time we left Africa was as Homo erectus. Those individuals dispersed and moved around and ended up becoming extinct due to whatever reason. They were outcompeted. And then Homo sapiens moved out a second time. So the Homo sapien species developed in Africa. So that is where the evolution of the whole species happened. And then we left and dispersed and did all of the, moving around and taking over that we enjoy doing. And when we look at mitochondrial DNA, going back to what we were just talking about, the mitochondrial evidence supports this theory that we can trace all of our genetics back to Africa. And how they do that is basically count up how many mutations there are in the DNA, and the group that has been around the longest will have the most mutations, because mutations take a long time to build up in a population. And so the longer you're around, the more likely you are to have more mutations. The less you've been around, the less mutations you're going to have in your genome. So when we're comparing different ethnic groups, the African ethnic groups have the highest amount of mutations compared to any other ethnic group on the planet. So that supports the fact that at one point in time, we all came from one spot. We were all in one area. And you guys can kind of take that as you need to, as you want to. You might be like, this is complete hooey, and that's totally your prerogative. You are allowed to say that. This is just the current theory based on the current evidence that we have in the scientific community. And then our last slide. I have one more, but it's a funny slide. It has no information. So one more thing we can use to try and track evolution is these things called interspersed elements. And these are very noticeable pieces of DNA that we can very easily see in a DNA strand that scientists think came from viral infections in our ancestors. So they think that at some point in time, an individual got what they refer to as a retrovirus, and viruses can insert their DNA into cells. That DNA can then be taken into that individual's DNA and just become a part of it. Doesn't necessarily do anything. It's just kind of sitting there. But we have the evidence that that viral infection happened, and we can kind of compare these elements between different species. And if we have the same kind of viral strains at around the same point in our DNA, scientists say that there must have been a common ancestor that we share that got infected with that virus before the speciation event occurred that caused us to change into different species. And so they're kind of using these interspersed elements to kind of try to see if they can figure out how we are all related to each other. And the two different kinds that we have are signs and lines. They're really, really simple. Signs are short interspersed elements that are about 300 bases long of just constant repetitive bases. So like AT, 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 AT for 300 bases. It's a lot and a little weird. And then we also have lines, which are long interspersed elements that are around 5,000 bases long. So AT, 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 AT for 5,000 letters. It's a very large chunk. And it's not usually in a coding area, which means it's not usually in an area that's going to turn into a protein. So it doesn't really do anything to the organism as a whole because it's not turning into anything. But they're very good evolutionary markers. So that is mutations and evolution. And I will leave you with this hilarious comic. This is a virus. Looks kind of COVID-y, not gonna lie, but this was made before, like way before COVID. I've used this picture since like 2014. So, little virus and a retrovirus who is rocking his afro and his handlebar mustache and using the awesome retro font, being like, hey, I'm awesome and retro. Way cooler than a regular virus. Still, I can laugh at myself even when you guys aren't here. So, haha, I'm hilarious. But that's all I have for you. If you read on Google Classroom, I posted what you are going to be working on now. You should have hopefully about half an hour-ish left of class. And you have your alien activities that are due tomorrow. I would probably focus on getting those done. Make sure you're getting the picture as well as the questions. There are questions that you need to hand in or else you will not pass this assignment. So please finish those. Your biotechnology questions that we got yesterday, those are due on Monday. You guys can start working on that. There's also a review package at the front of the room that you guys can take, and I recommend that you take and start working through this weekend to help study and review for your unit exam that is next Tuesday. And next Monday, we will have a review period to go over all of the wonderful things that we have talked about since September, the beginning of September. So that being said, no bio tomorrow. I will see you guys on Monday. Have a great weekend and a great rest of your day. And now I'm going to awkwardly try to stop my recording. And I will see you guys later. Bye. Nope, I didn't stop it. Now I'm stopping it. Okay, bye.